right, in this lesson, we're going to take a look at muscle contraction and the mechanics. The first thing we want to look at is something called the sliding filament theory. And what the sliding filament theory is it proposes that the filaments aren't shortening, but that they're actually passing by each other. And you've got two different types of filament groups that we're going to look at. You've got thin and thick. And the thin filaments are actin, troponin, and tropomyosin. And then your thick filament is myosin. And we're going to talk about that here in a second. But that's what I want you to remember, is that they don't shorten, they actually just pass by each other. And when you're looking at, we need to go over a couple of things here. So this is your A band right here. You got your I band, and you've got your M line, which is your midline, which is contained kind of within your H zone. And S actually stands for sarcomere, which is the functional unit that we're actually talking about here. And then Z-line, and your Z-lines are your edges of your sarcomere. And when your muscles get stretched out, think about it, we want to try to make this guy as long as possible. And what happens is that your Z-lines go out and they get further apart. Your I and H bands don't get longer. And then your A band doesn't change. Now, between stretch and maximally contracted, your A band isn't going to change at all. It's that you're going to have changes within your I and H band. So if we look at the other way and go the other way and maximally contract the muscle, what we're going to find is that your I and H bands essentially disappear, and your A band's the same, but your Z bands get closer. They go in, or Z lines get closer. So this is something I want you to think about when we're talking about the myofilaments as they actually shift past each other to create a shortening of the entire muscle cell. But before we do that, we have to understand how we get the impulse from the nervous system. So we're going to look at something called the action potential. What the action potential is it's basically the nerve impulse to whatever destination we're going to. So in this instance, what we're looking at is something called the neuromuscular junction, or something that I'll call later the NMJ. And when we're talking about the neuromuscular junction, we're talking about the nerve impulse going from the nerve to the muscle fiber. And this is how it happens. We need to look at some quick anatomy though. This is the infoot. This is the end of the axon. Then you have synaptic cleft, which is the gap. This is the space between the neuron and the muscle fiber. And then you've got the motor end plate, which is part of the sarcolemma, which is the cell membrane of the muscle. And what happens is we want to get the nerve, we want to continue. So you've got this gap. And so we basically want to say, hey, we need to continue this nerve impulse down from the nerve all the way to the muscle fiber to get it to do what we need to do. So this is how it happens. You get your action potential that comes down the nerve, and it hits this infoot where you have this influx of calcium, and this calcium enters the infoot and stimulates these are vesicles, and the vesicles stimulate the release of acetylcholine into the synaptic cleft. They kind of move down this way and attach, and then they release the acetylcholine across the synaptic cleft. So it basically bridges the gap here, and then the acetylcholine binds to these receptors in the motor end plate. And then what happens is you get when this happens, it promotes the action potential down the muscle fiber. And when that happens, it passes over the sarcolemma into the T-tubules, and then you get this influx in calcium, which is what we want, because calcium is required to go into the myofibrils and cause contraction. So what does that look like? Well, before we figure out what it looks like, we have some conditions that we have to meet. So the first thing we want to look at is something called the all or none principle. You cannot have one or two muscle cells contract with a nerve stimulus. Basically, all muscle cells contract or none at all. So that's something that you need to be mindful of. The other thing we need to think about is normal temperature. In order for optimal muscle contraction, you can't have excessively high or excessively low temperatures. Also, muscle contraction is not going to happen normally in the presence of fatigue substances, so things like carbon dioxide, lactic acid, or ADP. So what are the resting conditions that need to be in place before we actually have a muscle contraction? Well, a couple things need to happen. First off, your tropomyosin needs to be covering the active sites on the actin molecule. You'll see this in a second, but basically you have this thing that looks like this. This is your actin molecule. It looks double helical. And then you have this other thing, which is your tropomyosin. And in its normal spot, it covers the active sites. The other thing is you've got your myosin filament, your thick filament, and you've got your myosin head. And it has to have ADP plus an organic phosphate attached. It can't be ATP because that means it's in its resting position. That means it's all the way forward. What happens is when you have ATP attached, it pulls it back. It's like cocking a gun. The other thing that you have to have is low calcium in the sarcoplasm. If you have high calcium, you have a whole cascade that happens, which I'll explain in a minute. But when you have low calcium, that means it's resting. When you have the influx of calcium, that triggers the muscle contraction. So what does a muscle contraction look like? And we're going to show this in an animation in just a second, but I want to walk you through the process. First off, you have an action potential that releases calcium. You got to have calcium first. Then calcium binds to the troponin, and that causes the tropomyosin to expose the active sites on the actin filament. When that happens, the activated cross bridge combines with the exposed actin. What that essentially means is that the myosin then has a way to attach to the head, or that the myosin head attaches to the actin and then basically binds to it. And then the myosin deactivates and slides that process over, or slides the filament over, and that shortens the sarcomere. So we'll go through it again. You've got an influx in calcium. You have and it binds to the troponin. It causes the tropomyosin to expose those sites. Then you have the myosin cross bridge activation. And then when the myosin deactivates, it slides it over and it shortens the contraction or shortens the sarcomere, and that means you have a contraction. So then what happens? Well, ATP comes back and attaches back to that myosin cross bridge. With this part is a little bit counterintuitive because you think you need ATP for energy. Well, in this case, what happens is normally the myosin head is here with ADP. ADP plus the inorganic phosphate. But the problem is we need it to go backward. Well, it requires energy for that process to happen so that it can basically come back and move and push that filament forward again. So in order to do that, you need to get it in this position, and that requires ATP. So in relaxation, what happens is it comes and the ATP comes in and it moves this head down and it disconnects from the actin and reactivates it, and it returns it to this resting position. Calcium returns to the sarcoplasmic reticulum, so you get that decrease in the calcium. The tropomyosin goes back and recovers those active sites, and then you get the thin filaments returning to their resting position. So let's take a look on the animation to see what it looks like. So I know you're looking at this thing and going, oh, what is going on? All right, so let's start from the beginning and look at what we have. So you've got the myosin complex here, which is what we're looking at, which is this guy. Then you've got your actin helix and your tropomyosin. So this is your actin right down here. And then your blue that we're looking at is the tropomyosin. The troponin, which is important here, is the purple. And then you've got your calcium. So remember the process. So calcium comes in first. You have the action potential that says, hey, we need a muscle contraction. Calcium comes in and attaches. When it does, the troponin it activates the tropomyosin, which exposes the active sites. When that happens, then is kind of the next phase, which is you have to have all those things happen before you can even get myosin to do its thing. When that happens, the myosin is immediately attracted to it. Then you have this, if you look, you'll see the ATP, the green ball right here. It comes in, and it comes in when it does, the myosin head lifts up. So it lifts up, and that's because the ATP is basically putting back in that position to help slide it forward. So it slides it forward and then moves up and relaxes. You'll also see this, this little gray pyramid triangle thing comes in. And basically what that does is that helps to facilitate the process of converting ATP to ADP plus inorganic phosphate, which is required to be in that resting position. So I encourage you, it's a lot to study this process, to think about what we've talked about. Okay, so let's recap. Remember your contraction components. You need myosin, actin, troponin, tropomyosin. You also need calcium and ATP in order to make your contraction happen. Your myosin, this is your thick filament, and these other ones are your thin filaments. These make up your I band, and your thick filament is your A band. Remember, your action potential goes from the infoot of the axon to the motor plate via the synaptic cleft. And the reason it happens is because of acetylcholine. You get that acetylcholine that crosses it, and it continues the action potential down the neuromuscular junction, or across the neuromuscular junction, down the muscle fiber. You've got your normal contraction, and the things that you need for a normal contraction, don't forget this. You need your normal temperature, and you have to be absent of those fatigue substances like CO2, so carbon dioxide, ADP, and lactic acid. Remember that we're talking about the sliding filament theory, that the sarcomere shortens because the thin filament passes the thick filament. And then finally, ATP and calcium, just like we talked about here, are required to create motion from that myosin head and to reveal those active sites. I know this lesson was a lot. Thank you guys for hanging in there. Make sure you check out all the resources attached to this lesson. Now go out and be your best selves today. And as always, happy nursing.